Good evening, and thank you for having me. My name is Michael Zirpoli. I'm an aspiring science educator and mycologist. I am currently a scientific instructional technician at the Evergreen State College, from where I graduated and conduct my research. Over the past 3 years, I've been engaged in an assortment of research projects related to morel mushrooms. I've spent countless hours collecting, identifying, culturing, and attempting to cultivate morels from around Washington State. This is my presentation, Morel Hunting in Wisconsin. Again, my name is Michael Scott Zirpoli. Tonight, I'm going to present you with information related to hunting morels in Wisconsin. I will speak on basic identification, habitats morels can be found in, what species of morels grow in your area, and tips and tricks from my own time in the field. I've never hunted for morels in Wisconsin, but I have attempted to combine knowledge I have from my academic work and experience hunting morels elsewhere, as well as work of others more familiar with the area, to create an engaging and informative presentation. First, I'm going to take a step back and talk about what exactly a morel is and how it differs from other edible fungi and some similar inedible fungi. Morels are ascomycetes, one of 6 phyla of fungi. These fungi are the goody grabbers, fruiting early in the decay season, breaking down simple sugars. They often exhibit complex life cycles, including asexual stages. For mushrooms, ascomycetes typically form cup or sac-shaped fruit bodies, as well as hypogeous fungi like truffles. Morels are one of the few exceptions in terms of ascomycetes that produce large fruit bodies. In ascomycetes, spores are produced in 8-spored cells known as asci. In morels, this occurs in the pits. The mycelial structure and growth pattern of morels and other ascomycetes is different from that of basidiomycetes. Basidiomycetes, like Amanita muscaria, pictured here, are your more common cap and gill mushroom. I mention this phyla of fungi because the vast majority of mushrooms we hunt for food are basidiomycetes. Truffles and morels are among the few ascomycetes that are actually hunted for food. Morels belong to the family Morchellaceae, which also contains one of the false morels, which we'll talk more on here in a second, Verpa. In addition, there's a number of hypogeous fungi similar to true truffles, which are both edible and incredible. The Oregon brown and black truffle are both members of Morchellaceae. Another member of the family is Discotis, which is morphologically dissimilar to morels and more reminiscent of other cup fungi. If hunting for morels in the wild, it is important to be aware of inedible toxic species occurring in similar locations at similar times with similar appearance, namely Gyrometra and Verpa. I have primarily run into Gyrometra in montane regions in Washington, but I know it occurs in eastern North America in hardwood forest where true morels can be found. Another genus of concern is Verpa, which can be confused with the half-free morels. Aside from the 2 species of half-free morel, all other morels have caps that are fully connected to the stem and should be easy to differentiate from Verpa. The number one way to tell a true morel apart from a false morel is whether or not it's hollow. Most true morels have stems that are also completely connected to the cap, with the exception of the half-free morels, which do connect to the cap just further up. Verpa will be somewhat hollow, but when young, it is filled with a cottony pith. Gyrometra will not be hollow at all. The cap may appear so when cut, but the stem will always be solid. The cap of Gyrometra is also more irregular than that of true morels. Again, you often find morels and false morels occurring at the same time in the same place. And if you are hunting morels, please take the time to familiarize yourself with these other genera. True morels come in all shapes and sizes and are found in a variety of ecosystems across the world and are both edible and incredible. Morels have always been a source of taxonomic confusion, epitomizing the age-old argument of splitting and lumping, with some scientists recognizing as many as 30 species of morels, while others recognized as few as 3. Before the application of molecular taxonomy, there were almost 350 different proposed names for morels on Index Fungorum. This inability to build a functional taxonomy resulted in confusing research, highlighting inconsistent results, leading to more confusion surrounding morels. We used to delineate into groups like yellow, half-free, black, and these characteristics are not entirely helpful for actually telling morels apart. Differentiation was based largely on morphology, but as scientists have come to discover, the morel has a somewhat stagnant body plan with a surprising amount of morphological plasticity within a single species, which adds to the confusion. Needless to say, the old idea of IDing morels based solely on color is long gone. Within the past decade, the application of DNA analysis has revolutionized our understanding of morel diversity. Scientists have looked at multiple informative loci and used combined data from samples collected worldwide to establish basic phylogeny and evolutionary history of morels. Additional analysis of underrepresented regions has led to the discovery of additional species, and other work has been aimed at assigning either known names or new names to species identified in these studies. In total, phylogenetic analysis has identified at least 65 distinct species within Morchella and delineated 3 major lineages or clades. The Rufobrunia clade, which has 2 species characterized by red blushing and growth forms resembling that of both other morel clades. This clade represents the basal lineage of all other morels and appeared in the late Jurassic period in present-day Southern California. Only one other species is in this clade, and it's found in Turkey. Then there's the Alata clade, which are your black morels. This contains at least 36 species characterized by longitudinal primary ridges with secondary transectory ridges and a more regular pitting, and most commonly they're found with conifers. These morels are widespread, having first appeared in Western North America and having undergone several species radiation events across the globe. This clade also includes the half-free morels. The Esculente clade, which is the yellow morels, has at least 27 species characterized by haphazard ridging and pitting patterns and is more commonly found with hardwoods. This clade first appeared in eastern North America and, like the black morels, are widespread and have undergone species radiation events on more than one continent. Because of their evolutionary history, there are more black morel species in Western North America and more yellow morel species in Eastern North America. We now know what seemed to be a ubiquitous organism is actually a cast of species. Some morels are endemic and only appear in very limited areas, while others are more widely distributed. Multiple species fruit one after another in some areas, often with overlap, and sometimes they fruit in completely different environments. 30 of the 66 species identified by earlier studies have been assigned scientific names. Since then, more species have been assigned names by other researchers, and this progress in our understanding of morel diversity has kind of opened the door for a deeper comprehension of issues that have plagued the study of this mushroom for decades. I've been personally fascinated by morels because the more I learn about them, the more I realize we have so many unanswered questions about this mushroom, from their life cycle to the way they get their nutrients to the finer details of morel biology. These questions have evaded enthusiasts and scientists for decades. For such a popular mushroom, it's one of the most mysterious, and a lot of this mystery is driven by taxonomic confusion, which is why I feel it's important to have some familiarity with the species in your area. Until you know spots where morels grow, finding morels can kind of be a crapshoot. They seemingly grow both everywhere and nowhere at the same time, and the triggers of their fruiting are shrouded in mystery, and old wives' tales have long dictated our hunting patterns. The best way to find morels is to actually know which species grow in your area and, just kind of forget the old wives' tale and learn some taxonomy. But before I do talk about which species grow in your area, I want to give a general overview of when and where morels fruit in Wisconsin. Each Morel species has its own preferred habitat and time in the season when it fruits, but there are more general patterns that are worth noting. In the Midwest, morels emerge from late March through May and necessitate the convergence of several important environmental factors. Night temperatures must hover around 40 degrees Fahrenheit and day temperatures between 60 and 70, and the ground must reach between 50 and 60 degrees for the morels to fruit. In addition, ideal soil moisture levels must be met. Too much rain or too little in the morel season can be a complete bust. Needless to say, as is the case for many mushrooms that we hunt, some seasons are better than others, and abundance can differ from one site to another. Different species also prefer different temperatures, with some occurring earlier in the season while others can persist later into warmer weather. In the early spring, morels tend to grow along south-facing slopes. As the season progresses, they may be found along north-facing slopes. Many species are known to reoccur in the same location year after year, while there are other species that seem more prevalent after disturbance events such as fire, flooding, or tree death. There's a lot of prime hunting ground in an assortment of ecosystems found throughout Wisconsin. Most of Wisconsin's forest was cleared for timber and agriculture in the late 1800s, early 1900s. Northern Wisconsin's forests have come back, though few trees are more than 125 years old. Much of southern Wisconsin was developed in the farmland after the forests were cleared, and in those areas, the forests have only regrown in marginal crop and pasture land. Slowly, the state's forests have been coming back and maturing, and today about 46% of Wisconsin is forest. So that's about 16 million acres of forest in Wisconsin. The state is home to dozens of different hardwood species and a number of conifers as well as hundreds of understory plants, all of which play important roles in composing the environments in which morels grow. In Wisconsin, morels are most often hunted in undisturbed woodlands. Morels typically grow in sandy soils near ash, aspen, elm, and oak trees, but are found in a variety of other habitats in an association with a variety of other trees. Bottomland hardwood forests, dominated by green ash in the southern portion of the state, provide ideal hunting ground. These forests are found along rivers and streams. They're more common in southern Wisconsin, but are also found in the northern part of the state. The oak forest of southern and central Wisconsin are also good areas to hunt. Forests and even sandy dune areas near the Great Lakes also provides fertile hunting ground for one particular species of morel found in Wisconsin. Spanning 11 northern Wisconsin counties, the Chequamegon-Nicolet National Forest and Brule River State Forest are good examples of prime morel foraging territory in northern Wisconsin. One could expect to find both black and yellow morels in these woods, depending on the timing and area being hunted in. Pine barrens found across the state are also a great place to look for morels. Now I will take some time to cover the species found in Wisconsin. I will also cover a few species that are potentially in the state. And we're going to begin with the blonde or yellow morels. We're going to start with Morchella americana. This species has pale yellow ridges and pits that do not darken brown or black with maturity, but they can be dark gray when immature, as you can see in this far right picture. Its cap is attached to the stem without a significant groove or channel, a feature that's known as a sinus in morels. Among the yellow morels, it can be distinguished on the basis of its medium to large size, its egg-shaped cap, the random orientation of its pits and ridges, and the fact that its surfaces do not normally bruise reddish when fresh. In old age, it can sometimes discolor reddish-brown, but not actually bruising red when fresh. Morchella americana is possibly saprophytic and mycorrhizal at different points in its life cycle. It's known to grow alone scattered or gregariously in a variety of ecosystems. Morchella americana fruits in the spring, obviously, March through June, depending on the latitude and the elevation. Morchella americana is the most widely distributed common yellow morel in North America. In Western North America, Morchella americana primarily appears in river bottoms in association with cottonwoods and other hardwoods or in urban settings. I've only ever found it in an alleyway in my neighborhood and in a garden next to my house, which only has a single fruit tree planted in it and is surrounded by conifers. It's also known to grow in association with ornamental ash plantings or apple trees. East of the Rockies, it can essentially appear almost anywhere that there's trees, but it's especially frequent under dead or dying American elms and under living white ash and green ash. It's also found under apple trees in old untended orchards, non-commercial apple orchards from what I've heard, and occasionally under conifers, along old railroad beds, steep slopes. It's been reported in burn areas as well as, again, in riparian areas like we have it here on the West Coast. There's a name you may have heard, Morchella craspus, which has sometimes been applied to yellow morels with large inflated stems. Such collections are not supported by DNA testing as a separate species. Rather, they merely represent very mature specimens that have developed over long periods of time in warm, wet conditions. Morchella americana is one of the most common morels in Wisconsin. It's been reported far and wide in the state, especially in the southern hardwood forests, but also in the northern and central parts of the state. Unfortunately, DNA analysis is needed in your area to truly tell this morel apart from a very closely related species named Morchella ulmaria. Morchella ulmaria is found only in the Great Lakes region, from about Wisconsin to Ontario, all the way over to western Pennsylvania. When it's young, this morel has bald, flattened, pale yellowish ridges and yellowish to gray yellow pits. And when it's mature, it has sharpened to eroded yellow to brownish yellow ridges and similarly colored pits. Much like Morchella americana, Morchella ulmaria is possibly saprophytic and mycorrhizal at different points in its life cycle. It's known to grow alone, scattered, or gregariously under hardwoods. Much like Morchella americana, it associates with white ash and green elm, especially first year after elm death. So like we had just said, americana is fond of dead elms, so is ulmaria. This species typically fruits from April to June. In the Great Lakes region, you're kind of stuck not knowing whether you're collecting Morchella americana or Morchella ulmaria without testing their DNA. That's just kind of, unfortunately, the short of it. Of the two species, Morchella americana seems to be more common within the region, but Morchella ulmaria does occur frequently. We talk about commonality. That's obviously going to be based on DNA testing at this point in time. So it's hard to say which is the more common species. Researchers have found that there are no consistent morphological differences between ulmaria and americana. It's been suggested that maybe ulmaria is a paler species and its ridges are more flattened, but again, that's not always the case. Until we do more sampling, analyzing, and DNA analysis, we're not going to be able to suss out any actual morphological features that are useful for differentiation. Habitat doesn't appear to separate the two species either. Several times they've been found growing within just a few feet of each other. And it may just be the case that after more collections are made, that we do come up with characteristics that can be used to differentiate ulmaria from americana. But until then, again, this is a cryptic species pair that is going to necessitate DNA analysis to fully tell apart, which obviously can be kind of frustrating if all you want to do is hunt and eat the mushroom. But I suppose knowing the true name isn't the most important thing there. But if you're interested in collecting the mushroom for a barium sample or anything like that, you're definitely going to want to proceed with some DNA testing. Moving on, Morchella prava. This species looks like an irregular or deformed version of Morchella americana or Morchella ulmaria. Its pits are irregular in outline and in arrangement, and the mushroom generally has a gnarled-looking appearance. Its key differentiating feature is that it lacks sterile elements on the ridges that are present in ulmaria and americana, so not only can you use the macromorphology to tell these species apart, but also the micromorphology. When young, the species has bald or finely velvety, flattened or widely rounded, pale yellowish to whitish ridges. It's medium to dark gray or black pits. When it's mature, it has more bluntly rounded to sharp or eroded, brownish yellow to yellowish brown ridges and similarly colored pits. But it often can remain in its gray stage for prolonged periods of time and sometimes just stay gray for its entire life. Morchella prava is, again, like other Morel species, possibly saprophytic, possibly mycorrhizal at different points in its life. It's known to grow alone, scattered, or gregariously under various hardwoods and conifers. DNA collections of prava have come from a wide variety of habitats, including mountain conifer forests to filled-in swimming pools. But most of the collections have been made in sandy soils near rivers or lakes, under oaks and pines. The distribution of the species appears to be northern, from about Montana to Ontario, and it fruits April, May, and June. Morchella prava has been collected for many years in northern Michigan, along the Straits of Mackinac, often within a few feet of the water. In Wisconsin, it's been reported to grow along the banks of Lake Michigan, as well as north along the banks of Lake Superior. It's also been collected in the forests surrounding these lakes, as well as in sandy oak forests in the more central parts of the state, away from the lakes, more close to river in these cases. Morchella prava is closely related to Morchella dunensis, which is a species that grows in the Mediterranean in similar sandy environments. Dunensis grows in the sand dunes along the beach in the Mediterranean and is known to form dense mats of mycelium in the sand. It's been reported that dunensis actually tastes salty and fishy and briny, being that close to the water. But Morchella prava is actually, someone recently told me that it's the most delicious morel they've ever eaten. Our next yellow morel, which may or may not actually occur in Wisconsin, is Morchella diminutiva, which features that distinguish Morchella diminutiva from other yellow morels include its small size, the sparse vertical arrangement of the pits and ridges, its usually pointed cap, and its frequently long and skinny stem. When it's young, it has yellowish, rounded to flat, bald ridges in medium to dark gray pits, and when it's mature, it has sharpened or eroded, brownish yellow to yellowish brown ridges in similarly colored pits. Like other yellow morels, it features ridges that do not darken to brown or black with maturity, and a cap that attaches to the stem without much of a groove. Like all other morels, it is possibly saprophytic and mycorrhizal at different points in its life cycle, growing alone, scattered, or gregariously under hardwoods from April to May. So this is an earlier occurring species. Morchella diminutiva appears to be widely distributed and common in eastern North America from about the Great Lakes southward. It's fond of white ash, green ash, tulip trees especially. It's commonly referred to as the tulip morel, and hickories, although it can be found under oaks and other hardwoods as well. especially old apple trees, and it's not known to be associated with conifers at all. And it has not been officially reported in Wisconsin, but the southern part of the state is pretty prime territory for its occurrence. The species is particularly fond of tulip trees, but again, it's known to associate with other trees. The tulip tree does not occur natively in Wisconsin, but all these other trees that it's known to associate with do occur in Wisconsin. So there's definitely a possibility that the species could be found in the southern parts of the state. It has a close lookalike called Morchella virginiana, but that species is usually larger and more egg-shaped and appears to be exclusively associated with tulip trees in the southeastern United States. So you should not be seeing that species in your area since it's exclusively associated with tulip trees. Occasional specimens of Morchella diminutiva can look a lot like Morchella americana, but typically the cap is less conical and the arrangement of pits and ridges is less clearly sparse and vertical as opposed to the randomly arranged pits of Morchella americana. Moving on to the black morels, we start with Morchella angusticeps, which is the most common and widespread black morel of eastern North America. Like other black morels, it has ridges that darken with maturity, pits and ridges that are primarily vertically oriented, which is often referred to as being like a little raceway for ants. Morchella angusticeps also has a cap that is attached to the stem with a small but noticeable groove. This is a good example of the sinus that I've been mentioning. The species, possibly saprophytic and mycorrhizal at different points in its life cycle. It's known to grow alone, scattered, or gregariously under hardwoods, including white ash, cherry, poplars, green ash, tulip tree, and pines. And this species occurs a little earlier, starting in March, and grows through May. Morchella angusticeps is generally the first true morel to appear in the spring in eastern North America. It fruits 2 to 4 weeks before Morchella americana and the other yellow morels that appear in the same areas. In Wisconsin, angusticeps can be found in the southern and northern hardwood forests, as well as conifer-dominated areas. Morchella angusticeps is widely distributed east of the Rocky Mountains. Throughout most of its range, it's the only occurring black morel and is thus easily identified. Only in the northern part of its range is there a lookalike species, north roughly of the 44th parallel. It should be compared with another species called Morchella septentrionalis, which we will talk about now. Morchella septentrionalis has a cap that is conical, bluntly conical, or with a rounded apex, pointed and ridged, with its pits primarily arranged vertically. When young, it has a finely velvety or nearly bald, flattened, tan to brown ridges and brownish pits. When mature, it has flattened to sharpened or eroded black ridges and brown to yellowish brown pits, and it's attached to the stem with a small sinus. Septentrionalis is possibly saprophytic and mycorrhizal at different points in its life cycle and appears under hardwoods, including big-toothed aspen, white ash, and it's found usually growing near woody debris or growing directly from rotten hardwoods. And it's only been reported in May north of about the 44th parallel, occurring from about Wisconsin to New York. Septentrionalis is very similar to the more widely distributed Morchella angusticeps, but Septentrionalis is smaller than Morchella angusticeps, and it also has smaller spores. Additionally, it tends to appear in the vicinity of or even fruiting from the decaying wood of big-toothed aspens and other hardwoods. Large specimens of Septentrionalis might be confused with smaller specimens of angusticeps. In that case, using microscopy to compare the spores is going to be your best bet for positive identification. At the very least, it is possible to tell these species apart without DNA analysis. You might have to use a microscope, but based on its habitat, its morphology, and its spores, you should be able to differentiate it. It's not been officially reported in Wisconsin, but it has great potential to be present in the state. The big-tooth aspen is one of Wisconsin's fastest growing natives, and it can be found throughout the state in an assortment of environments. So it's definitely worth keeping an eye out for this species. Based on its more northern distribution, northern hardwood forest may be the prime location for hunting in the state. As you can see here on Mushroom Observer, it's been reported right over the border in northern Michigan. Half-free morels have caps that are attached about halfway down the stem, which creates a substantial overhang where the margin of the caps hang around the stem. We have two species in North America, which were referred to as one species at a point in time, but DNA analysis has supported that what was thought of as one species is actually two genetically distinct species here in North America, known as punctipes and Fungal Phyla. And though the two species cannot be separated on the basis of their physical features, they can be separated by geography. Morchella punctipes appears in hardwood forests from the Great Plains eastward, while Morchella Phyla appears under black cottonwoods in the northwestern North America. Morchella punctipes has a cap that is conical or broadly conical. It's pitted and ridged, with the pits primarily arranged vertically. When it's young, it has bald, flat, and yellowish-brown to pale brownish ridges, with whitish to pale yellowish pits. And when it's mature, it develops flattened to sharpened dark brown to black ridges and yellowish-brown to brownish pits. And it's attached to the stem in a skirt-like manner about halfway from the top. And again, the half-free morels are completely hollow, unlike easily confused verpa. Morchella punctipes is possibly saprophytic and mycorrhizal at different points in its life cycle. It's known to grow alone, scattered, or gregariously under hardwood in various forest types. It's common some years and rare or absent in others. And it occurs March to May and is widely distributed east of the Rocky Mountains. This species typically fruits before other morels, usually at the same time as verpa, and in similar environments. And due to the half-free nature of the species cap, it can be easily confused with verpa. So again, you want to make sure you take the time to learn the difference between these species. And always remember, if you have doubts about the mushroom you're looking at, just throw it away. In Wisconsin, Morchella punctipes is one of the most commonly reported morels, especially in the southern and central forests. And from what I've heard, it's one of the most delightful morels to eat. Moving on, Morchella importuna is commonly known as the landscape morel. It's a black morel that's easily distinguished on the basis of its habitat and its vertical primary pitting pattern that's common in other black morels. Morchella importuna has a cap that's conical or nearly so, and it can be occasionally egg-shaped or round. It's pitted and ridged with distinctly deep vertical channels with regular ladder-like cross ridges. It's definitely one of the more elaborate black morel caps. It has a lot of pits and ridges. When it's young, it has bald or finely velvety, blunted, rounded, grayish to brownish ridges in similar colored pits. And when it's mature, it has sharpened or eroded dark brown to black ridges and brown to olive or yellowish brown pits. And it's attached to the stem with a sinus. Morchella importuna appears to be limited to landscaping sites, gardens, planters, and so on. In North America, it's fairly common in the Northwest, where it appears in urban settings in the late winter and early spring. But the reason I'm mentioning it here is because it has been reported rarely and without DNA verification from locations in the Midwest and Eastern North America. So there is a distinct possibility that the species is occurring in landscape sites in Wisconsin. Morchella importuna is one of the few species that's clearly saprophytic when it's growing in treeless environments. It could be potentially mycorrhizal in other locations, as it has been reported occurring in post-disturbance environments in Western North America. And because it's clearly capable of a saprophytic life cycle or lifestyle, it's one of the few morels that's actively cultivated. In China, there's a massive outdoor industry that's been continuing to grow over the past decade, and Morchella importuna is the primary species being used in this industry. Moving on, in Western North America, yearly conifer fires produce hulls of morels large enough to sustain global demand. The morels growing in these recently burned forests are distinct species. And you may or you may not know that morels in Eastern North America have been reported to occur in recently burned forests. Recent studies have identified one particular burned species, Morchella exuberans, occurring in northern jack pine forests in Michigan, as well as in Tennessee and the Smoky Mountains. Morchella exuberans is one of 3 frustratingly similar morel species that appear in Western North American conifer burn sites. Morchella exuberans has some physical features that can be used to separate it from these other species, including the fact that it has a stem that is often chambered and layered internally, and it has elements on its sterile ridges under the microscope that are referred to as capitate, which mean they're roundish or swollen at the end. It should be noted that just about any morel can actually develop a thickened chambered stem, like Morchella crispus, as we mentioned before, if it's left growing unpicked in favorable growing conditions. But the stems of exuberans are all chambered even when young. It's possibly saprophytic, possibly mycorrhizal at different points in its life cycle, and it appears in lightly burned spruce and fir forests in the summer or spring immediately following the fire, and only that first year following the fire. And considering that there's one burned species possibly in Wisconsin, it's possible that the other burned species are there as well. On the basis of present data, Morchella exemia cannot be separated from Morchella sextulata without the use of DNA testing. Both of these species appear in burned conifer forests in Western North America, primarily in the spring or summer following the fire, but sometimes in dwindling numbers for a few springs afterwards. Both species are black morels, which means that their ridges darken with maturity, and their pits and ridges are primarily and vertically oriented, and their caps are attached to the stem with a small noticeable groove. No consistent differences in physical features have been observed between exemia and sextulata with or without a microscope, so DNA analysis is the only way to tell these two species apart. And though color appears to not be a reliable way to separate the burned morels on our continent, there is one species that is the exception to this rule. Morchella tomentosa, as you can see here, is incredibly dark with young age and loses some of that pigment with maturity, but the use of a microscope will show you, even in the most mature samples, that the pigmentation remains in the cells through the development of this mushroom. So again, this is actually one morel that you can tell apart based on color. The rest of the potential fire species, if you were to find burn sites in Wisconsin, would necessitate DNA analysis to tell apart. Michigan's DNR releases yearly reports of potential morel spots for the coming seasons, which is largely based on prescribed burning from the previous year. Unfortunately, the same is not done in Wisconsin, but one can easily access burn records online, going back as far as 6 years on the state's DNR website. In Wisconsin, these species are likely to occur in pine forests which have been recently burned, so the northern part of the state is definitely the best spot to look for potential burn morels. Now that I've covered the species of morels in Wisconsin, I want to take some time sharing tips and tricks I've learned hunting for morels. One way to increase your chance of finding morels in the wild is taking time to understand them on a more scientific level. For example, I've just been showing knowing a morel by its species name can give insight into why you might be finding it, where and when you are finding it, which increases your chances of finding it again. Morel hunters have traditionally stressed the importance of particular trees occurring in the areas where they are finding morels. This idea has led many to suppose potential mycorrhizal relationships, although this state of nutrient acquisition has not been definitively proven in a natural setting. More than anything, this habit of paying attention to the occurrence of trees can be translated to looking at a broader ecosystem pattern, taking into account not just one tree species, but all of the species occurring where you are finding morels, as well as the conditions during, before, and after fruiting. Another useful trick is knowing how morels reproduce, because it is not simple and includes intermediate steps, which can be useful in the identification of potential morel hunting ground. Something that makes understanding and hunting morels so difficult is confusion surrounding some of the basic biology of the fungi. Things like their life cycle and trophic statuses are still in question to this day. There have been a number of life cycle proposals for morels over the years, with the first being presented in 1990 by Volk and Leonard. This proposal has served as the basis for all future proposals and identified key steps in the life of a morel, which include the formation of primary and secondary mycelium, formation and germination of pseudosclerotia, and the development of fruit bodies. This proposal was nonspecific and said to apply to all species of morels. Another life cycle proposed in 2014 attempted to integrate all the known research about morels and expanded the life cycle to include asexual stages, and both proposals are still considered theoretical to this day. One other life cycle proposal worth mentioning was from 2007 and attempted to integrate conflicting reports concerning the trophic status of morels and also included some ecological conditions under which the life cycle takes place. There's evidence supporting numerable nutrient acquisition modes in morels, ranging from mycorrhizal to saprophytic. It has even been proposed that morels may switch between trophic modes throughout their lives. Confusion about this topic has been precipitated by taxonomic misidentification of morels studied in previous research. Needless to say, the topic of morel life cycles is not a closed case. More research is needed to develop our understanding of these fungi. Formation of pseudosclerotia is a response to unfavorable environmental conditions and also a strategy that fungi use for nutrient storage. Morel pseudosclerotia are also believed to enhance survival over winter, but the conditions that trigger fruit body formation arising from pseudosclerotia are not clearly understood. They're an orange to yellowy mass that can be found in the natural setting, often before the fruiting of morels, and they're especially commonly observed in burn areas. It's thought that pseudosclerotia can be produced from both primary and secondary mycelium. Factors affecting mycelial growth and pseudosclerotia formation of some morels have been investigated, but the results and conclusions have not been consistent due to the different strains and experimental designs. In addition, pseudosclerotia are not always formed under experimental conditions, and the ability and requirements of pseudosclerotia development varies greatly depending on the species. But we do know that all species observed at this point seem to produce pseudosclerotia. Another important phase of the life cycle of morels that can be used in your hunting is asexual reproduction. Much like pseudosclerotia, the exact role of asexual reproduction in the life cycle of morel is not entirely clear. One asexual stage was first identified and reported in 1904. In attempts to cultivate morels on apple compost, a contaminant was identified and named Constantinella. What was thought to be a contaminant was isolated and failed to germinate on media. And years later, DNA analysis showed us what had been named Constantinella and thought to be a contaminant was actually the asexual form of the morel species being worked with. More recently, scientists have directly linked asexual stages of morels to sexual stages using DNA analysis. And although we do not know the exact role of the asexual phase in the natural setting, we do know that it occurs in the natural setting and it can be used as an identifying feature for potential morel spots. Massive blooms of this powdery mildew have been reported in successful indoor morel cultivation. It's been observed in abundance in burn sites before and during morel fruitings, and it's also been reported in outdoor field sites in China where morels are cultivated. And in the cultivation of morels, it's considered to be a necessary stage in their life cycle. And again, we don't know the exact role of this powdery mildew in the totality of the life cycle of the morel, but we know it's important and we know that we can use it to our advantage in identifying morels in the field. I myself have observed asexual stages of morels in attempts to cultivate the fungus, as well as in the field before and during sexual fruiting. These are just some examples of Constantinella in natural settings. Some are associated with spots that produce morels yearly, while others were spots that appeared after I had inoculated the soil with Morel sclerotia. But you can see just this fine powdery mildew can be observed as early as the fall before a morel fruiting. So if you see something like this, it might be worth revisiting a spot the following spring because it could potentially be a morel spot. It's worth noting that morels take time to develop, upwards of 20 days. Unlike other mushrooms, they don't just pop up overnight. Development can be a very sensitive time, and drastic changes in environmental conditions can easily lead to the death of mushrooms. So it's important to keep an eye on the weather and plan accordingly. I also want to mention that the morel season is probably longer than you think. It's worth keep looking in spots before and after normal times that you would find morels. It's worth looking in spots nearby with more shade or more sunlight if you're looking earlier in the year. Move up in elevation if possible. In the early season, look in more sun-exposed spots in areas further south. Even within a few hundred yards, morel fruiting times can be as different as a month. So it's something you definitely want to be persistent about. Well, despite being so popular, we still have a lot of unanswered questions about morels, from their life cycles to the preferred reproductive modes to the way they get their food. It seems like every time we answer a question about morels, we find ourselves asking at least 100 more. Someone once told me that more morels are picked for the dinner table than for the herbarium, and I myself have struggled to put samples into storage as opposed to my belly, but as I've learned in my work, there's a huge value in taking the time to appreciate morels as more than just food. In doing so, it can create a more informed hunt, and it can put you in a position to progress our understanding of this mushroom. So take a moment to appreciate all that morels have to offer aside from just their delicious flavor. And with that, I will say goodnight, and I would like to thank the Wisconsin Mycological Society for having me and allowing me this opportunity to speak. I would like to thank my mentor, Paul Przybylowicz, Mike Beug, Carrie O'Donnell, Lolita Calabria, all of the science staff at the Evergreen State College, generous grants from the Puget Sound Mycological Society, and my former research partner, Heidi Steinbach. Thank you and good night.